Hi there. In this video, I'll be talking about bacterial genomes, and then towards the end, a little bit about archaeal genomes. So here are the distributions of sizes of genomes for bacteria and archaea. And again, these dark regions indicate free-living bacteria and archaea, and the white regions on these distributions indicate parasitic ones or endosymbionts. And you'll notice that they sort of cross over with the sizes of viruses as well as organelles. So in general, I'm talking about genome sizes that are between about 300,000 or 200,000 up to about 10 million base pairs. In this case, for double-stranded DNA now is all we're going to be talking about from now on. So this is a circular phylogram where everything over on this side, those are bacteria, and then in the middle here on the left are archaea, and sort of in the northwest corner here are the eukaryotes. The first bacterium I'm going to talk about is E. coli strain K12, which is the standard strain that lives in the guts of many mammal species, including humans. First, a little bit about what E. coli does. So this is E. coli K12. It's a gamma proteobacterium. The genome size is just a little over 4.6 million base pairs, and it has almost 4,300 genes, including 7 ribosomal RNA gene operons. And I told you that's sort of the minimum number for being able to divide every 20 minutes. And then 86 tRNA genes, a lot of tRNA genes. And remember, there's only 20 amino acids here we're talking about. So there are multiple tRNA genes for some of the amino acids at least. So E. coli is actually a species complex consisting of many species. So this is just K12. And if we had 0157, that's a pathogenic E. coli. So it's a facultative anaerobe. That is, it mainly grows in anaerobic environments, such as the human gut and other guts. But it can grow out in oxygen, so we can grow it in the lab as well. It's commonly found in the lower intestines of warm-blooded animals or organisms. It colonizes infants within 40 hours of birth, which I always thought was pretty amazing. So its optimal growth is at 37 degrees centigrade, which is human temperatures. But it's related to a pathogen, Shigella, a related genus. So it can live for a short time outside of the body, but it needs that temperature mainly. The oxygen isn't as much of a concern. It can live on a wide variety of substrates, so it can ferment sugars to produce lactate, succinate, ethanol acetate, carbon dioxide, and a small amount of hydrogen gas. It can also oxidize pyruvic acid, formic acid, hydrogen, and amino acids. And it can reduce oxygen to form water, nitrate, dimethyl sulfoxide, trimethylamine N oxide. It does have flagella, which requires a lot of genes, and it can undergo conjugation. That is, transfer DNA from one E. coli cell to another E. coli cell. And it or the species complex, diverged from Salmonella more than 100 million years ago, about the time there was a separation between reptiles and birds and mammals. And so you got this Salmonella growing primarily in reptiles and birds, and E. coli growing primarily in mammals. So then, regarding the genome, if you can grow anaerobically and aerobically, you need different sets of gene products, and you need to recognize when you're in a non-oxygen situation and an oxygen situation, and switch on or transcribe and express the proper genes and proteins for that. You also have to have proteins that are most active at those temperatures. They can live outside of the body for a short time. So that means both temperature and oxygen are different. So they have to be able to react to that, requiring more genes and signals. Can live on a variety of substrates. So it needs many genes to grow on different substrates and to produce different compounds. And also the oxidation and reduction reactions here require specific enzymes as well. So that's another group of genes that are needed for those processes. And then flagella are produced by a multi-gene family consisting of dozens of genes. So it needs that set of genes. It also needs a set of genes that allows for conjugation to recognize the other E. coli cell and attach to it and then exchange DNA. So all of those processes need genes to produce the products to do each one of those processes. So here's a little bit about E. coli. They are rod-shaped bacteria requiring a certain set of genes to do that. There's an inner membrane and an outer membrane, and the DNA and ribosomes are inside, the cytosol in the middle. And then it has flagella coming off of the side. So each one of those requires sets of genes to produce each one of those parts of the cell. Now, as I said, they can grow aerobically or anaerobically. These are ones that are growing aerobically. So it can switch from one set of genes to another set so that it can survive in oxygen. Some strains also can infect vegetables, and then those can infect humans when they eat those vegetables or handle the vegetables. So that's another group within this species complex. So here's what I mean by species complex. So here's E. coli K12 as well as another strain, and you can eat those, drink those, and you'll be just fine. In fact, in many ways, they aid digestion, and they actually produce amino acids and things for you when you have them in your gut. Some other E. colis here, 0157, are deadly. They can kill you. So they're related in some way here along this tree, but a little further away. And then there are other E. colis down here that infect urinary tract and kidneys, as well as some that are avian. Now, although they can inhabit each one of those tissues and organisms, they're not necessarily disease-causing E. coli. As I said, 0157 can be deadly, can cause serious disease, and it's related to Shigella more closely than to E. coli K12. So when you talk about E. coli, it really is a species complex, and not all of them are related in the same way. So it's probably in some cases, the names don't really tell you exactly what the organism is. And up on the upper part here, there's more Shigella. And again, those are more closely related to K12. Those cause serious disease and then appear as Salmonella. And so back here, about 100 million years ago, there was this split between Salmonella and these E. colis and Shigellas. Again, these inhabit mainly reptiles, and these down here inhabit birds, of course, but also humans. Also, another factor here, as I showed you earlier, K12 has about 4,300 genes. But if you look at all of the E. coli together, then together it includes about 16,000 different genes, also indicating the diversity within that species, which, appears to be more than one species. So here's a diagram of the complete genome as it was sequenced just around 1998, 1999, somewhere in there. So 4,639, 221 base pairs in the genome of K12. If you look around this colored circle out here, All of the things in yellow are genes that are on one of the strands of the DNA, and then the ones in orange are on the opposite strand of the DNA. So the genes are fairly well packed on that genome. This next circle in, these little green arrows, whitish green arrows, those are where the tRNA genes are. So you can see they're dispersed throughout the genome. And then these red little arrows there, those are the ribosomal RNA loci. So you can see also they're pretty much dispersed around the E. coli genome. And in E. coli, there's only one origin of replication, and then it proceeds in both directions around here. And the terminus for replication is over here. The sort of star pattern in the middle here, originally they looked at whether there were any GC and AT rich regions. So that's all that that central part is. I won't discuss that much. It's not as important for our discussion. So here's a table of some functional groups of protein encoding genes. So about 4% is devoted to regulatory sequences, and then there are cell structures, membrane proteins, structural proteins, phage, and viruses, transposons, plasmids, etc., running down the line. And when I first saw this, the first thing that I and many others noted is that about 38% of the genome contain genes of unknown function. So E. coli has been studied as a genetic organism. Mutants have been made since the 1950s, I think, when it was first started as a sort of genetic lab strain that could be dissected genetically. But still, when they sequenced it, 38% of the genome had genes in it that were completely unknown and not predicted. And then another high percentage, 87 genes, 2.3 of the genome, are viruses, transposons, and plasmids. So a fairly large proportion is made up of these. And by the time we get to humans, I'll show you that 8% of our genome come from viruses, originate from viruses. And then about almost 200 genes are involved in cell structure, that is, making the cell wall, making the membrane proteins, etc. Makes up about 4.5% of the total genome. So the group of genes that are responsible for gene expression primarily, that is DNA recombination, replication, repair, etc., transcription, translation, nucleic acid biosynthesis, amino acid biosynthesis, fatty acid biosynthesis, these central pathways and biosynthetic pathways, about 600 genes making up more than 13% of the genome. So quite a large proportion are devoted to these processes. Here are two pie charts, one showing a general classified versus unclassified or unknown as far as the types of genes. In this case, for this analysis, about 48% were classified, and about 51% were unclassified. So a large portion, 780 genes, 18% of the genome, are involved in central metabolism. And then, again, you have this DNA replication, transcription, amino acid production, protein secretion, etc., making up a fairly large proportion of the genome. And then building the cell envelope. Here are some genes specific to translation, fatty acid metabolism, and then transposons, viruses, etc. These percentages vary somewhat from the previous table that I showed you because they're from different studies will look at genes in a slightly different way, so you can get variation that way. And this is true of many of the studies. So some will make a gene call, that is, identify a gene using one set of criteria, and a different group might put that in a slightly different category. And some genes and gene products have double duty. That is, they're acting in several different pathways. And so depending on the categorization, you can get some changes in where those genes wind up in the categorizations. So that's E. coli. It's actually a fairly complex bacterium, not the most complex, but probably in the top 25% as far as complexity. It can live in several different environments. It can live on many different carbon sources. It has a capability to live at slightly different temperatures. So that's one reason for its complexity. It's fairly diverse in the things that it can do. So the next one I want to talk about, Aquifex aeolicus, is simpler. It has a very narrow ecological range, that is, environmental range. It can only live in certain places, that is, in very hot water. And outside of that, it can't. So it has actually fewer genes and fewer capabilities to adapt to different locations. So here's an Aquifex aeolicus cell. It's a rod-shaped bacterium with flagella, so it does have those genes. But it grows in very hot water, and the water doesn't change temperature very much during the day or night. It's constantly hot water, and it was first isolated from some hot water pools in Yellowstone National Park. So here's a little bit of information about the genome and how this organism lives. It has a genome size of about 1.5 to 1.6 million nucleotides or base pairs. So it's about 1/3 the size of the E. coli genome. Also has about 1/3 as many genes, 1,522. In one study, as I told you, when people make calls, they come up with different numbers. One study reported 1,778 genes. But anyway, either one included 1,728 encoded proteins, 6 to 7 ribosomal RNA operons, and that agrees in both studies, and 44 tRNA genes, which also agrees in both studies. So this could probably, with 6 to 7 ribosomal operons, divide as rapidly as once every 20 to 30 minutes. It's among the oldest extant bacterial lineages, so it's been its relatives and ancestors have been on Earth for a very long time, probably in excess of 3 billion years. And a lot of scientists agree that probably one of the first places that bacteria and organisms in general evolved was in hot pools, either on the surface or in deep ocean, around deep ocean vents. So it is thermophilic, and strictly thermophilic. It can't grow at in cold temperatures. And its proteins are thermostable, which you would expect. It grows, its optimal growth is 85 to 95 degrees centigrade, which is really hot. So sometime if you have 85 degree water, even 65 degree water, will burn you. So this 85 to 95 is extremely hot water. They're aerobic, although they can live under low oxygen conditions by reducing sulfur instead of oxygen. And some members are anaerobic, and they reduce nitrogen instead. Then it reduces oxygen to form water, which is clear. And then approximately 16% of its genome appears to be close to Archaea. And so these things live in close proximity to Archaea, certain members of thermophilic Archaea. So it's not surprising that they have exchanged genes along the way. It is a chemolithotroph, lithoautotroph. So it can fix carbon from CO2, but it's not photosynthetic. So it does this by using other chemicals to get energy from those to fix the carbon onto other carbon compounds. The enzymes used in aerobic respiration are similar to the enzymes used in anaerobic bacteria. So it appears that these have probably evolved first as anaerobes and then transitioned to become aerobes by some mutations causing differences in some of their enzymes, so they can use those enzymes under aerobic conditions as well as anaerobic conditions too, or low oxygen conditions. The next bacterium I want to discuss, or group of bacteria, are cyanobacteria, and I'll talk mainly about this guy here, Synechococcus. Synechococcus has a genome size of about 2.5 million base pairs, about 2,800 genes. So it's smaller than E. coli, which, initially you might think, well, it has those photosynthesis genes and pathways, so it should have more, but it doesn't. It's not able to live under as many conditions as E. coli, so it has a smaller genome. 2 are RNA copies, gene copies, so it actually can only divide once every hour or so at maximal rate. And 44 tRNA genes. Remember, E. coli had about twice that many. So it's a photoautotroph. It fixes carbon dioxide using photosynthesis in the Calvin cycle. And in photosynthesis, it has photosystems 1 and 2. At least most cyanobacteria do. This one does have both photosystems 1 and 2. So it splits water into oxygen gas, electrons, and protons. And the protons, excited by photons of light, are shuttled through these protein complexes and into the thylakoid lumen. The electrons, the electron energy is used to move the protons, and then the protons ultimately are shuttled through ATP synthase to make ATP. And the electrons and protons are added to NADP to make NADPH or NADPH2. And then ATP and NADPH2 are used in the Calvin cycle to produce carbon compounds, to fix the carbon. And the 2 photosystems actually evolved separately in 2 different lines of organisms, and then came together in Cyanobacterium. So that's a completely different story. But these 2 photosystems have significantly different protein complexes in them. So these are gram-negative. That is, they have 2 membranes, and picoplankton. So they're small cells, and they account for 25% of the organic production in oceans. So they're pretty important out in the oceans, especially, although the original cyanobacteria appear to have evolved first in fresh water and then moved to salt water. They can use nitrate, ammonia, and in some cases urea as sole nitrogen sources. They have phycobiliproteins, phycocyanin, allophycocyanin, etc. These are proteins that hold on to the chlorophylls. Phycobilins are also found in red algae, but not in green algae. They've evolved past that. And also in some other photosynthetic organisms. These are marine, primarily, occurring in all oceans, although limited in the polar regions. They do not like cold water. Genetically variable, so Synechococcus in general has genomes all the way from about 2.4 million base pairs up to 3 million base pairs. Here's a little bit on the morphology of cyanobacteria. If you take a cross-section across here, you can see the DNA inside condensed into the nucleoid, and ribosomes are in there, and then the 2 membranes out here. And then you have these thylakoid membranes and phycobilisomes coating those thylakoid membranes. So up here you can see one of those cut down this a cross section of those, and the phycobilisomes are here, and those are essentially antennae that will gather in the light and excite the bacterial chlorophylls, which will then transfer that energy down into the photosystem 1 and photosystem 2. And here's a comparison between what happens in red algae and then what you see in green algae in plants. And this one's just showing photosystem 2, but you'd have photosystem 1 also. So here are the phycobilisomes here, which then collect that light energy and funnel that energy down to this special bacterial chlorophyll down here, which then excites electrons to break water into oxygen and the electrons, and then those are moved down the electron transport chain. In green algae and plants, what you have is, instead of phycobilisomes, there is a light harvesting complex that does about the same as a phycobilisome does. So similar, and then this one shows photosystem 2 as well as photosystem 1, and you can see that they contain very different sets of proteins, and they evolved in 2 different types of bacteria before coming together in cyanobacteria and other gram-negative photosynthetic bacteria. In the lower part of the figure, it's indicating some of the gene clusters that each one of these has. It's not all of the gene clusters. There are many others involved in making that entire photosystem work, including constructing each one of the photosystems, but also in producing the various bacterial chlorophylls that are needed in these processes. So there are biochemical pathways for those as well. So just to remind you of these different pathways, then we have the light reactions here, where you gather in light in both photosystem 1 and photosystem 2. This one breaks water into oxygen and protons, and the electrons from that reaction also flow through here. They're excited again in photosystem 1 and then added to NADP to form NADPH. And some of the electron energy also is used to pump protons into the lumen of the thylakoids. Those then flow through the ATP synthase to make ATP. And then over here in the dark reactions of photosynthesis, Rubisco fixes CO2 utilizing the NADPH and the ATP in further processes to produce more carbon compounds. And here are a few more details, including the phycobilisomes here that are present in cyanobacteria and red algae, which then hold those cytochromes that react with the light, energize those cytochromes, send that energy down into chlorophyll in the photosystem 2, which then breaks water into protons and oxygen and electrons, and electrons flow through the electron transport chain in the membrane, the thylakoid membrane. And some of that electron energy is used to pump protons into the lumen of the thylakoid. Those protons then flow through the ATP synthase to make ATP out in the stroma, and that ATP is used in the Calvin cycle. And the electrons ultimately wind up on NADPH, and that is also used in the Calvin cycle. So just generally speaking, that takes a lot of genes and gene products to accomplish photosynthesis, including all of the proteins in the light reactions, those in the dark reactions, production of the organelle itself, and production of the thylakoid membrane itself. So a lot of different things are necessary to make photosynthesis work in these cells. So here's the Synechococcus genome. This is the one that's 3 million base pairs in length. And all of these on these 2 outer rings, those are genes on one strand of the DNA, and then The other circle is the genes, or are the genes in on the other strand. And as with the other two that I've talked about, especially E. coli, you can see that the genes are very packed in these on the chromosomes of these organisms. So unlike eukaryotes that we'll see later, these genes are really packed next to one another. So here's are the gene categories for Synechococcus, the one that's 2.4 million base pairs in length. Almost 60% have been classified, and about 40% or more are unclassified. Here are the photosynthesis genes, only making up about 4.2% of the total genes in the genome, which initially surprised me, but it's about 1/3 as many that are used for central metabolism. So that's still, a specialized function of these, about 4.2%. And then there's transcription, there's the cell envelope, so a higher proportion of genes for the cell envelope here, probably because there are multiple membranes. Then translation, DNA replication, amino acid synthesis, and several other processes in this genome. The last bacterium I want to talk about is this planktomycete, Pyralula, which planktomycetes have internal membrane-bound organelles, including in Gemeta, the genus Gemeta, an organelle that looks a lot like a nucleus. So they are unusual in that way. They also have fairly large genomes. So I'll talk about that a little bit. So on the right-hand side, this is Pyralula, and I'll talk about its genome. But you can see some of the internal membrane-bound organelles. They also bud. When they divide, it looks like budding, like in a fungus in some of these. And then Gemeta here, which has a double membrane around what looks like a nucleus, and that encloses the nucleoid where all of the DNA is, and they also have a lot of ribosomes in there as well. And some have hypothesized that this internal membrane structure was the source of the nucleus in eukaryotes, as well as possibly some other organelles, membrane-bound organelles. So here's a little information about Pyralula. Its genome is over 7 million base pairs long, which is about 1.5 to 1.6 times the size of an E. coli genome. And it has over 7,700 genes, many of them involved in maintenance and construction of internal membranes. So you can imagine why that is. Each compartment that you set up needs a different set of enzymes and proteins and membrane components to make those compartments functional in each of their processes that are going on in those different organelles or different compartments. So this bacterium is a freshwater spherical bacterium. It can be anaerobic or aerobic, so it needs different sets of genes to do those. Most genes are for replication, transcription, metabolism, are closest to those of bacteria. But then many of its genes are more similar to eukaryotic sequences, which sort of ties them in with being the ancestor of eukaryotes as well, or at the very least a major contributor to genes in eukaryotes. So multi-copy genes also make up more than 25% of the genome, and that's more consistent with eukaryotes than prokaryotes or bacteria. They're chemoheterotrophs, so they're not autotrophic. They have no peptidoglycan in their cell walls. Their ribosomes are attached to that nuclear membrane, or that nucleus-like structure. And again, that's similar to what you find in eukaryotes, including ribosomes attached to the rough ER, which is a membrane that's continuous with the nuclear membrane in eukaryotes. And the nuclear membrane is continuous with the cell membrane, and this is similar to what you find in eukaryotes also. They have polar yeast-like division that is budding, so it looks like a fungal cell dividing. Has a large number of sulfatases, of which 75% are bacterial-like and 25% are eukaryotic-like. And these are used to scavenge carbon from sulfonated carbon compounds. As I said, it's not an autotroph, so it needs to find a way to get carbon compounds, and this is one of the ways that it uses. They have a large number of transposons, 81, and although there are transposons in other bacteria, this is a large number for a bacterium. It also has FTS genes and some genes similar to tubulins, which are found in spirochetes, which are a related group, but then are found mainly in eukaryotes, alpha and beta tubulins. It has the highest number of membrane targeting sequences of any bacterial group, and this is probably because you have so many membranes, so many locations, that you need those signal sequences or target sequences to send those proteins to the correct membrane, correct location in the cell. So here is the genome laid out in several circles. So these outer two circles indicate the genes on one strand versus the other strand. So you can see they're quite packed together. And of particular note, you can see quite a few genes involved in protein synthesis and destination, as well as those involved in transport. And you might expect those in an organism that has many membrane-bound organelles within it. The next circle indicates alien genes, those that came from elsewhere, and highly expressed sequences. And then the next circle indicates those sequences that are closer to those from Archaea and eukaryotes. Again, indicating that there have been gene transfers probably into this organism from Archaea. The ones that are eukaryotic-like, they're probably eukaryotic-like because these genes were contributed to the original eukaryotes 2.7 billion years ago. The next circle primarily looking at those sulfatases, a large number of sulfatases in yellow, and those are the ones that are able to scavenge carbon from sulfonated carbon compounds. The next circle indicates transposons. As I said, there's a large number of those, integrases, and then evidence of lateral gene transfer or horizontal gene transfer. These are transfers from one species to another, so you can see there are some of those as well. And again, the total genome size is just over 7 million base pairs with about 7,700 genes, so a lot of genes. And in fact, if you add together an archaeal genome with one of these planktomycete genomes, the total approximates the size of a small eukaryotic genome. So some people have hypothesized that this was the source of the nucleus going into an archaeal cell that then became the first eukaryotic cell, and then later a mitochondrion or an alpha proteobacterium joined those to become the first mitochondrial eukaryote. So the next thing I want to talk about is archaeal genomes, and today I really only have one to talk about from an Archaeum. So here they are, the archaeal genomes. They have about the same distribution as bacteria. I think bacteria look broader because more of them have been sequenced, but eventually I think the Archaea might have just about as much genomic diversity. So the one I want to talk about is Methanococcus yonashii. And if you ever look this up as far as the genome paper. It was called Methanococcus at that time, but it's been reassigned to the genus Methanococcus. And the first thing to note is that Archaea are over here with eukaryotes, and they are separated to a lesser degree than both of those are from bacteria. And I'll show you in a few minutes another piece of evidence that shows that eukaryotes and Archaea are not as different as they each are from bacteria. As you might guess from the name, Methanococcus is a methanogen, and it grows around hot thermal vents, deep sea thermal vents, and this is one of those. So it was isolated from a thermal vent and then sequenced. But you can see they're pretty much spherical bacteria with a lot of flagella, and that's primarily so that they can maintain a position in the water that is optimal for nutrition and temperature. And this particular thermal vent, these are known as black smokers, and the black color is often caused by high amounts of sulfur compounds. And those sometimes can be important for autotrophs. So here's the genome size for this Archaeum. 1.7 million base pairs, rather small, but it has a narrow ecological or environmental range. 1,796 genes, 2 ribosomal RNA operons, 37 tRNA genes, plus it has a couple of extra chromosomal elements, 58,000 base pair circular DNA, and with 46 genes, and that's about the size of a lambda bacteriophage, by the way, and then a 16,550 base pair extra chromosomal element with 12 genes, and that's more the size of what you would think a plasmid might be. Its core genes are similar to those of Haemophilus influenzae, but it grows at a higher temperature, so they are thermostable versions, having a higher GC content in the genome and more cysteines to form disulfide bridges to hold the proteins together. Many of the genes are more similar to eukaryotes than bacteria, which sort of sets where they are evolutionarily. That is, they're closer to eukaryotes than bacteria, and that is because the Archaea are the ancestral group to the eukaryotes. Temperature range 48 to 94 degrees, optimal 85, so they grow in high temperatures. It's a barotroph. It lives at high pressure on the ocean floor near thermal vents. It's a methanogen and a chemoautotroph and reduces CO2 with hydrogen to produce methane, so that's how it gets its carbon compounds. And it contains many, but not all genes for nitrogen fixation. So it's probably cooperating or collaborating with other bacteria and probably Archaea near those thermal vents to get some of these compounds it needs. It does contain histones, so these do contain histones to wrap up the DNA. And the genes are sort of midway between bacteria and eukarya, although they're actually closer to eukaryotes. Here's the genome laid out, about 1.7 million base pairs, and then there's the genes on one strand versus the genes on the other strand. So again, a fairly compact genome. These little H-looking things on the interior here, this was one of the first organisms that had its genome sequenced because it was a relatively small genome. But at that time, they had to break this up into clones. So each one of those represents a little clone of DNA, a little piece of DNA that was isolated, actually tens of thousands of base pairs, and given to someone in the lab, a graduate student, and it was spread out over several labs. And then eventually they put all this together into one large genome. There were places that were difficult to sequence, and those were sequenced in a slightly different way to get through those regions, a slower way actually. And some of these other lines in the center represent where the tRNA genes, ribosomal RNA genes, and a few other categories of genes are located. So here's the genome laid out in gene categories. And again, about 41% has been classified and over 50% is not classified. So energy metabolism, about 10%. Methanogenesis, which is how it's then getting its carbon compounds, 7.8%. And then you have translation, transcription, DNA replication, etc., making up a significant portion of the genome. Part of the reason is that the systems for transcription, translation, DNA replication are more complex than in bacteria, and those actually carry over into eukaryotes later as well. And you can see that more clearly here. So this diagram shows you bacterial type genes in red, eukaryotic genes in blue, and then what archaea have. And you can see they're sort of intermediate between bacteria and eukaryotes. So up here you see these genes up here, ribosomal, total number of genes, ribosomal operons, cell division proteins. These all are pretty much bacterial-like because most archaea are unicellular. So they take on that unicellular type activity and function. But they also originated about 500 million years or so after bacteria, so they've had more of a time to evolve. And so their systems for doing translation, ribosomal proteins, translation, elongation, and initiation factors, then the transcription factors, DNA polymerization, replication, things like that, are more advanced. That is, they're more complex than those in bacteria, and they have more similarities to those found in eukaryotes. So this is another indication that they've had more time to evolve, so they're a little more complex, and that these systems in archaea led to the systems that you find in eukaryotes. Then you can see this again here. There's less distance between archaea and eukaryotes because an archaeal member, or ancient archaeum, actually gave rise to the eukaryote. So it was one of the two in the endosymbiotic event that led to the eukaryotes. The other one was probably a planktomycete, and then later an alpha proteobacterium, and then after that a cyanobacterium to become the plants. So through these genomic studies, you can not only determine some of the things that are characteristic of each one of those species, but they will tell you a lot about the evolution of these organisms and even the complex organisms. This is a more detailed phylogram that was done a few years ago with a large number of sequences, a very large number of sequences, and these are the bacteria here. These bacteria here actually are have never been studied in a lab. The only way we know that that whole group exists is from their sequences. So there's a whole group of bacteria that are known only from their sequences. And so you can do whole genome sequencing sometimes on things that you can't really grow or look at in the lab. And then down here you see the archaea, and off of a basal branch of the archaea, you see the eukaryotes coming out of there. And so the eukaryotes were based on archaea with a few bacteria entering that eukaryotic line from time to time to form the nucleus first of all, and then the mitochondria next, and then the chloroplast next, and then other organelles in the protists as well. However, the last ones in the protist group are really eukaryotes moving inside of other eukaryotes, mainly to become autotrophic, photoautotrophic. But then you have some of those that have a eukaryote inside of a eukaryote also being enveloped by another eukaryote. So there are many different levels of moving those genomes from one species to another as an entire genome rather than just a few genes at a time. And I will talk more about that in the next video. But that's it for today. So thanks very much. I will talk to you later.